Today we are making competent cells of DH5 alpha bacteria, E. coli strain of DH5 alpha. Before doing anything, we need to clean the workbench and hood with 70% ethanol. This is our primary culture. I have inoculated in yesterday from DH5 alpha plate. Single colony was picked and inoculated. And this is our LB. This is 100 ml LB and this is primary. So I am adding just 1% of primary in whole secondary. Now we will grow it till 40.4 to 0.6. So this is shaker incubator where I am keeping it, and it will grow at 37 degree and RPM is 200 RPM. Now we have to check the OD, and OD should be from 0.4 to 0.6. This is our blank. We have to take and check the optical density of cells at 600 nanometers. This is OD 600. Initially we will set the blank. After that we have to check the OD of our cells. This is 0.6, which is fine. Now we are keeping the cells set in ice for 1 hour. After that we will further proceed. Now incubation in ice. I am transferring the cells, distributing the cells equally in 2 falcons. Whole process should be done at 4 degree. Now after transferring the cells into 2 falcon tubes, I am going to harvest the cell by using the centrifuge, and as you can see that the temperature of the centrifuge is already reached to 4 degree, and speed we are using 4000 for 10 minutes. So we have to keep the centrifuge tubes in the opposite direction. Now start. So now it's done. So this is the resuspension solution of 80 millimolar magnesium chloride plus 20 millimolar calcium chloride. This solution I am using to resuspend the cell pellet, and we will keep the cell pellet after resuspending in this solution for 1 hour in ice. Before resuspending the pellet, I am removing the supernatant from the tube. This is the cell pellet. This is a very crucial step. We need to be little bit careful in this step, because while resuspending the pellets, we do not take the tube outside the ice. So initially we will add 1 ml resuspension solution to the tube, and then we'll resuspending the pellet by moving the solution through the walls, not directly on the pellet. Same with the other tube. And this resuspension solution is ice chilled. We have to keep it at 4 degree before doing this process. Now I have resuspended the pellet with 1 ml resuspension solution. Now I am adding this solution up to 25 ml in each tube. incubate at this step for 1 hour in ice. So after 1 hour incubation in ice, I'm again harvesting the cell at 4 degree centrifuge at the speed of 5000 rpm for 10 minutes. After that, we will resuspend the cells and make aliquot. This is the final spin of whole process. These are the harvested cells. Now I'm removing the supernatant from each tubes. I have removed complete supernatants from each tubes, and meanwhile I have pre-chilled the tubes, almost 20 to 25 kind of tubes, and these are the this is 1 molar calcium chloride, this is H2O water, and this is 30% glycerol. These all we need in this process. Initially I will resuspend the pellet of 1 tube, and I'm going to add 1 ml 30% glycerol. microliter 200 microliter 1 molar calcium chloride, so that its final concentration will be 100 millimolar. These all things are pre-chilled and ice cold. Finally, to make the total volume 2 ml, I am going to make up the remaining volume with H2O, and I am adding 800 microliter water to the tube. Again, same, while resuspending the pellet, be little extra careful. We do not directly pour the solution over the pellet. Always place the tube over ice, never remove the tube from ice. Whole process should be done in ice. Tube 1 pellet was resuspended. I'm transferring whole solution resuspended cell to the second tube. This has been resuspended in the tubes, and now I am going to alicot 100 microliter in each tubes, and then we will freeze it at -80.